So we generalize this problem we just practiced into any gene pool with 2 alleles, and we call the allele frequency of big A being P and the allele frequency of little a being Q. So as long as our gene pool does not change, like this is the same bunch of alleles with only random mating going on, after 1 generation of random mating, we always produce 3 types of genotypes because we have the chance of drawing 2 alleles being either one of them. So since we only have 2 alleles in total, P plus Q always equal to 1. 1 means 100%. So similarly, since we have only 3 types of genotypes, if we add their frequency together, we have P squared plus 2PQ plus Q squared equals 1. So this is the equation called Hardy-Weinberg equilibrium. What this is telling us is random mating themselves maintains the variation. Variation does not get lost simply because individuals are mating with each other. And after 1 generation of random mating, you always get the same genotype frequencies being P squared for big A, 2PQ for big A, little a, and Q squared for little a. And it will stay like this as long as this gene pool does not change. So for this equilibrium to be maintained, we actually need this gene pool to literally not change, which means that for Hardy-Weinberg equilibrium to stand true, we need a lot of really stringent assumptions. So one of them is we want our system to be diploid so that each individual will carry 2 copies of the same thing. And we want random mating going on. And also to maintain that this gene pool does not change, first we need to assume that we have a large enough population so that even if we draw 1 allele from it, the allele frequencies don't really change. And also we don't want new things to come to this population. So usually there are 2 ways new things can happen to a population. Either by having an immigrant, like a migrant, like coming from a different population carrying new things, or having some de novo mutations, like just those alleles themselves started to change. So we don't want either of them to happen. We want a closed population and with no new mutations. Also, we want these genotypes to be completely neutral, meaning that each individual has an equal chance to survive. So we don't really want natural selection to favor one or the other genotypes. Otherwise, that will break the balance. And so given all these assumptions, it's probably not hard to imagine that in the real world, it's very hard for us to meet all of these assumptions at the same time. So we will always have a violation for these Hardy-Weinberg assumptions. So for example, in reality, nobody actually random mate depending on your preference. So there's always non-random mating going on. And we always have mutations going on in the DNA sequence, even if the chance is pretty low. And we have migration going on. And if we forget about the entities of these individuals, migration is essentially these alleles or genes flowing between populations. So we also call that gene flow. There's natural selection often going on in populations. And also there's populations always are finite in size. So depending on the specific population size, you may actually change the allele frequencies in each of the generations. We call that a genetic drift effect. In the next module, we're going to go into the details of those. So long story short, with all of these 4 mechanisms that are highlighted in blue, they essentially do one thing, which is to change allele frequencies. So it's like we have a closed gene pool, but since some things can be going on there, this gene pool no longer maintains the same, like over generations. So we call this evolutionary mechanisms or evolutionary processes, and all of them will change allele frequencies. So we actually define evolution as we see actual allele frequency change in space and in time in populations. With one exception of non-random mating, and soon we'll see what is going on with non-random mating. So there are a few implications with Hardy-Weinberg equilibrium. So one thing is, since we almost never meet all of the assumptions with Hardy-Weinberg equilibrium, it is hard to see that being exactly maintained in a real-world population. However, if we look at some of the DNA markers across the genome, we see that even though it's hard to meet all of the assumptions, most of the time we see a lot of these neutral markers being approximate to what we observe or what we expected to see from Hardy-Weinberg equilibrium. Overall, Hardy-Weinberg equilibrium describes a fundamental model for the populations, which is how variation is maintained by random mating. And most importantly, we only need one generation of random mating for the population to reach Hardy-Weinberg equilibrium. And as long as the allele frequencies don't change, it will stay that way. So, and now you have seen pretty much everything we're going to talk about in population genetics. So what exactly is population genetics? It is an extension of Mendelian genetics going from individual level to population level. And what we're interested in is essentially the variation patterns in the population, how it is maintained, and why it changes over time. So the specific way of how we study it is we need some variation data. It could be DNA variation data or RNA variation data. It could be any of those. And what we actually do is we build some statistical models to describe how these evolutionary forces, all four of them together or some of them specific combinations, actually change allele frequencies over time. So population genetics is a very mighty tool that can help us understand a lot of real-world questions, and which is why I do research in this. So some of the questions we're interested in that PopGen can help us answer includes ancestry and individual identification. So in California, this is especially the case because people will have ancestors coming from all over the world, and we're interested in to what extent we're still maintaining the variations from the original populations. So PopGen can help us characterize that and figure out the ancestry among each individual. Does anyone know who this guy is? This is a Neanderthal individual. This is an extinct human species and no longer existing on Earth. But once in the past, we have met each other. So we have discovered a lot of fossils for Neanderthals in Europe and also in some part of Asia. We can learn some things about them, but not everything. Fortunately, we actually we recovered some of their genomes from these fossil records. And from this, the DNA sequence, we learn more about the population history of Neanderthals and also how we might have encountered each other in our human past. PopGen can also help us figure out the phenotype variations and the genes that are linked to those and why this variation exists. And also in multiple disciplines of studies, we're interested in different types of disease, either in human or in other organisms. So you can use PopGen tools to help us figure out which genes are related to this disease, how the disease evolved over time, and overall that can help us to prevent and control disease more precisely.